Okay, so now let us study the folding of embryo and let us see what changes occur due to the folding of embryo. So, uh, firstly, there is cephalic fold. Okay, so there is cephalic fold of the embryo, then there is caudal fold of the embryo, and we have the two lateral foldings. So, we have cephalocaudal folding and lateral folding. Now, why the embryo folds? Why there is folding of embryo? The reason is first we have the cephalocaudal foldings. So the cephalocaudal folding is caused by rapid growth of central nervous system. Initially we had a neural plate, soon it enlarges to form the three brain vesicles, and the caudal part also gets elongated to form the spinal cord. So as the neural tube enlarges to form the central nervous system, there will be increased or rapid longitudinal growth of the embryo, and that's why we have the cephalocaudal foldings. Then there are two lateral folds. So why the lateral foldings are there? So lateral folding is caused mainly by the rapidly growing somites. The paraxial mesoderm will form the somites, and as the somites are rapidly growing during fourth week of intrauterine life, there will be formation of lateral folding. Okay, so we have the cephalocaudal foldings. So this is cephalic folding, this is the caudal folding due to the rapid growth of central nervous system, and we have two lateral foldings, and these are due to rapid growth of somites. Now what changes are occurring? Here you can see the yolk sac. There are two cavities. There is yolk sac. So this is the yolk sac, and this one is the amniotic cavity. So you can see the growth of yolk sac is retarded. Okay, so the growth of yolk sac is retarded, whereas the amniotic cavity will go on enlarging. It will enlarge to such an extent that it will enclose the entire embryo. Now the embryo is floating in the amniotic fluid. Okay, so the yolk sac is retarded and the amniotic cavity is enlarged. And we can also see that the cephalic fold, caudal fold, and the lateral fold, these all foldings are converging on the ventral surface. This is the ventral surface of embryo. So all these folds are converging on the ventral surface of the embryo, and the convergence is to such an extent that you can see this is the amniotic cavity, this is the amniotic membrane. So this is the amniotic membrane. So amniotic membrane is forming a tubular sheath around the connecting stalk. Okay, so the connecting stalk is enclosed by a tubular sheath, and soon it is converted to umbilical cord, and this yolk sac undergoes hourglass constriction. You can see the constriction here in the yolk sac. It undergoes hourglass constriction. Part of the yolk sac which is incorporated within the embryonic fold, this intraembryonic part of the yolk sac, it is giving rise to primitive gut, and the extra embryonic part of the yolk sac, its growth is retarded and it is known as definitive yolk sac, or it is also known as umbilical vesicle. Okay, so the intra embryonic part of yolk sac forms primitive gut, and the extra embryonic part of the yolk sac is known as definitive yolk sac, or it is also known as umbilical vesicle, and both the parts, intraembryonic and extraembryonic part, are connected with each other by vitalo-intestinal duct. So this vitalo-intestinal duct normally obliterates, but if it does not obliterate and if there is communication of primitive gut with the umbilical vesicle, then the condition is known as Meckel's diverticulum. Okay, now we will see what changes are occurring in the head fold. Now this is the head fold. Most cranially, at the cranial extremity, you can see the forebrain vesicle is there. Okay, so we have the forebrain vesicle, then we can see the cranial end of the notochord, then we can see the foregut. Okay, the primitive gut is divided into foregut, midgut and hindgut. So the part of primitive gut which is lying in the head fold is the foregut. Okay, so this is the foregut. Then you can see the stomatodium. This stomodium is the oral cavity. It is bounded cranially by forebrain vesicle. It is bounded caudally by the pericardial sac, and the floor of the stomodium is formed by buccopharyngeal membrane. Now the buccopharyngeal membrane ruptures at 4th week of intrauterine life, and the stomatodium or the oral cavity gains access with the primitive gut. Okay, so this is the buccopharyngeal membrane. After that you can see the pericardial sac. So ventrally we have the pericardial sac, and the pericardial sac is separated from the foregut by the cardiogenic area or the cardiogenic plate. Okay, so we have the pericardial sac and the cardiogenic plate. After that we have the septum transversum. Okay, so this is the septum transversum. So in septum transversum, two things should be known here. First is the derivatives of septum transversum and how a partition is formed which separates the thorax from abdomen. So this septum transversum was initially lying cranial most in the embryo, but now it has come to the ventral aspect and caudal to the pericardial sac. So uh, pars hepatica or the developing liver bud, it will divide the septum transversum in two parts. Okay, now see, this is the foregut, this is the midgut. So at the junction of this along the second part of the duodenum, the pars hepatica develops, means the liver is developing. It will then invade the septum transversum and it will divide the septum transversum in two parts. So the ventral part of septum transversum is known as pars diaphragmatica, and the dorsal part of septum transversum is known as pars mesenterica. Okay, so the ventral part will give rise to the anteromedian part of the diaphragm or the central tendon of diaphragm and fibrous pericardium, whereas the dorsal part will form the stroma of the liver. It will form the falciform ligament and coronary ligament of the liver, and it will form the lesser omentum. I will revise again. See, pars hepatica is the liver bud, developing liver bud. It will invade the septum transversum. It will divide the septum transversum in two parts. So the ventral part is pars diaphragmatica. It has two derivatives. First is the central tendon or anteromedian part of the diaphragm, and the second derivative is fibrous pericardium. Whereas the dorsal part, it will give rise to the stroma of the liver, then the falciform ligament and coronary ligament of the liver, and the lesser omentum of the stomach. Now let us see what changes are occurring with the formation of the head fold. So here you can see most cranially we had septum transversum, but now it has come caudally and to the ventral aspect. As the foldings are converging ventrally, you can see the septum transversum has come ventrally. Then if you see the pericardial sac was lying dorsally and the cardiogenic area was lying ventrally, but with the folding the cardiogenic plate has gone to the dorsal aspect and the pericardial sac is now to the ventral side. Okay, initially pericardial sac was present dorsally, but with the formation of head fold the sac has come to the ventral aspect. Then the stomatodium, it was present initially caudal to the cardiogenic area, but now it has come to the cranial part. Okay, it is cranial to the cardiogenic area, or it is present now in between the forebrain vesicle and the pericardial sac. Again, the important thing to be known about septum transversum is that we know that the intraembryonic coelom is there, and it gives rise to three cavities. The three cavities are pericardial cavity, pleural cavity, and peritoneal cavity. So initially there is a communication between the pleural cavity and peritoneal cavity through pleuroperitoneal canal. But this septum transversum will also help to form pleuroperitoneal membrane, which will separate the thorax from the abdomen, or which will separate the pleural cavity from the peritoneal cavity. So this is all about head fold. Now let us see the tail fold. Okay, so the tail fold, again the part of the primitive gut which is incorporated within the tail fold is known as hind gut. Okay, so we have the hind gut, then the connecting stalk and allentoenteric diverticulum. Now with the formation of tail fold will lie to the ventral aspect. Then this allentoenteric diverticulum, it will divide the hindgut in two parts, pre-allentoic part and post-allentoic part. So the post-allentoic part is also known as endodermal cloaca. Okay, so the post-allentoic part is known as endodermal cloaca. Now what is the fate of this allentoenteric canal? Its proximal part will get incorporated in the endodermal cloaca, whereas the distal part of this allentoenteric diverticulum will form urachus. Now this urachus extends from the apex of urinary bladder up to the umbilical cord, up to the umbilicus. So it extends from the apex of the urinary bladder to the umbilicus, and it will get obliterated to form median umbilical ligament. So when you are doing dissection of the anti-abdominal wall in infra-umbilical part, you can see in the median plane there is median umbilical ligament. So this is nothing but the remnant of urachus, and urachus is nothing but the distal part of the allentoenteric diverticulum. Okay, after that you can see an area called cloaca. So in the cranial part we have buccopharyngeal membrane. Similarly in the caudal part we have the cloaca, where only two germ layers are there, ectoderm and endoderm is there. There is no mesoderm. So this cloaca with the formation of urorectal septum will divide in two parts. So the distal or the posterior part is known as anal membrane, which on rupture gains access of the anal canal with exterior, and the ventral part is known as urogenital membrane. So it roughly ruptures by 7th week. Okay, and also the primitive streak, you can see here, this is the primitive node and this is the primitive streak. So the cells from the primitive streak will start migrating in the infraumbilical part, and these cells will give rise to the infraumbilical part of the ventral wall of the abdomen. Okay, so this is all about the tail fold. So what are the important things to be known? So along the tail fold you can see the distal part of the neural tube, you can see the distal part of the notochord, you can see the primitive streak, then the hindgut is present, then you can see the allentoenteric diverticulum, then the connecting stalk is there, you can see the cloacal membrane. Okay. Now let us see the changes occurring due to the formation of lateral fold. So what is the reason why this lateral fold is formed? So the lateral fold is mainly formed because there is overgrowing of the central part of the germinal disc. Means as there is rapid growth of the somites in the central part, it will give rise to two lateral folds. There is right and left lateral fold. Now the formation of lateral fold will give rise to some changes in the embryo. So what are the changes? Mainly the amnioectodermal junction will come to lie on the umbilical cord. So in the region of the umbilical cord, you have the amnioectodermal junction. Then the formation of the midgut is there. We know that the part of the yolk sac incorporated within the embryonic fold is forming the midgut. Now in the head fold, the part of primitive gut is foregut. In the tail fold, the part of primitive gut is known as the hindgut, whereas with the formation of the lateral folds, the part of the primitive gut incorporated is known as midgut. So this is the midgut. The vitalointestinal duct is communicating the midgut with the umbilical vesicle. Means intraembryonic part of the yolk sac is forming midgut, and the extraembryonic part of the yolk sac is forming umbilical vesicle, and the communication between these two is known as vitalointestinal duct. Okay, then you can see the paraxial mesoderm gives rise to the somite. Somite has formed primitive vertebrae. So this is the derivative of paraxial mesoderm. Then you have intermediate mesoderm, which gives rise to the kidneys and sex gonads. And then we have the lateral plate mesoderm. So intraembryonic coelom develops in the lateral plate mesoderm. So this is the splanchnopluric layer of the lateral plate, and this is the somatopleuric layer. So inner aspect of the body wall is covered by the somatopleuric layer. It gives rise to the parietal layer of all the three serous membranes, pleura, pericardium and peritoneum. So the parietal layer is formed by the somatopleuric layer. Splanchnopluric layer will form the visceral layer of all the three serous membrane. Also you can see here a double layer of mesentery is formed. So this is the dorsal mesentery. The dorsal mesentery is suspending the primitive gut, and this is formed by the splanchnopluric mesoderm. So the splanchnopluric intraembryonic mesoderm gives rise to the dorsal mesentery. And also you can see here the this is the intraembryonic coelom. So intraembryonic coelom is continuing, or it is communicating with the extraembryonic coelom in the region surrounding the umbilical cord. Now later on, with the completion of the lateral folds, and now they are converging towards the ventral aspect. So they are forming the tubular sheath around the connecting stalk, and now the connecting stalk is converted into umbilical cord. So you can see the umbilical cord is formed here with the convergence, ventral convergence of the lateral fold. Also you can see that the communication was there initially in between the midgut and umbilical vesicle. So this communication was known as vitalointestinal duct. Here the vitalointestinal duct has obliterated completely, and now there is no communication between midgut and umbilical vesicle. Okay, so again, what structures you can see here? There is formation of the vertebrae. You can see the dorsal aorta. Here also there are two dorsal aortas. So the dorsal aortas are seen, the midgut is seen. Then you can see the dorsal mesentery suspending the midgut, and here this is the primitive dorsal mesentery, dorsal aorta. Then the primitive vertebrae, intermediate mesoderm is there. Then this one is the splanchnopluric layer. This is the somatopleuric layer. This is the intraembryonic mesoderm. Sorry, intraembryonic coelom. Okay, so this is all lateral plate mesoderm with the intraembryonic coelom. So these are the changes occurring in the embryo with the formation of lateral fold. Main change is there is a communication between the midgut and umbilical vesicle through vitalointestinal duct, which will then obliterate. The communication is lost. Then the second important thing is the amnioectodermal junction is formed, so that the connecting stalk is now converted to umbilical cord, and the third important change is dorsal mesentery is formed by the splanchnopluric layer of the lateral plate mesoderm. Okay, and there is a communication between intraembryonic and extraembryonic coelom. So the endoderm differentiates into three parts. So now we can see this is the primitive gut, and the primitive gut is formed by the incorporation of yolk sac within the embryonic fold. So the intra-embryonic part of the yolk sac gives rise to the primitive gut. It is endodermal in origin, and this endoderm now differentiates into, first is foregut. So the foregut part of primitive gut is lying in the head fold. Then there is midgut. So this is the midgut. So midgut is the part of yolk sac which is communicating with the umbilical vesicle through the vitelline duct. So this is the midgut, and in the tail fold we have the hindgut. Thus the primitive gut is divided in three parts. There is foregut, then there is midgut, and there is hindgut. So the foregut is supplied by celiac artery. All the structures which develop from the foregut are supplied by celiac artery. Then the derivatives of midgut are supplied by superior mesenteric artery, and the derivatives of hindgut are supplied by inferior mesenteric artery. So all these three arteries, celiac trunk, superior mesenteric artery and inferior mesenteric artery, are the ventral branches of aorta. Now let us see the derivatives. So first the derivatives of the foregut. So in this region, in the ventral aspect, we also have the pharyngeal arch. So from the pharyngeal arch again has clefts derived from ectoderm, arches derived from mesoderm, and pouches derived from endoderm. So from the endodermal pouches of the pharyngeal apparatus develops the parenchyma of tonsil, thyroid, parathyroid, thymus. Then the liver and pancreas also develop from the foregut. At the second part of the duodenum, pars hepatica, uh, endodermal bud will develop into the liver, and for the pancreas we have ventral bud as well as dorsal bud, and these will fuse and they will form the pancreas. So pancreas is also endodermal in origin. Now, if we see this part of a primitive gut, it is giving rise to the lining of pharynx, then the epithelial lining of esophagus, stomach, and duodenum. So this is the duodenum. So in the second part of the duodenum, there is major duodenal papillae. So it is known as ampulla of Vater. So up to the ampulla of Vater is the foregut part, or the duodenum is partly developing from foregut and partly it is developing from midgut. So at the ampulla, up to the ampulla of Vater, it is developing from the foregut. Also the mucous membrane of the tongue develops from the foregut, and in the foregut along the ventral aspect we have the laryngotracheal groove from which the respiratory system develops. Then the auditory tube and tympanic cavity also develops from foregut. Okay, so these are the derivatives of foregut. From the branchial apparatus, from the branchial pouches, the tonsil, thyroid, parathyroid, thymus will develop. In the second part of the duodenum, we have the pars hepatica from which the liver develops, and ventral dorsal bud fuse to form the pancreas. And ventral to the foregut, we have laryngotracheal groove, and it will lead to the formation of respiratory system, auditory tube and tympanic cavity. Mainly it's epithelial lining. Then the primitive gut gives rise to the epithelial lining of pharynx, esophagus, stomach, duodenum up to ampulla of Vater, and the mucous membrane of tongue. Again, one thing to be noted here is the epithelial lining is only endodermal in origin. The musculature is mainly developing from splanchnopluric mesoderm. The connective tissue and the stroma is also developing from splanchnopluric mesoderm. Only the epithelial lining is endodermal in origin. Now, this is the midgut. So, the mucous membrane of the midgut. So, we have seen the foregut will end here at the ampulla of Vater. So from the ampulla of Vater of the duodenum, then the third and fourth part of the duodenum, then jejunum, ileum, then the cecum, ascending colon, and the transverse colon up to its junction with the left one third is derived from midgut. Means from the second part of duodenum, from the ampulla of Vater to the junction of right two third and left one third of a transverse colon will develop from midgut. Now the hindgut derivatives are mucous membrane of the gut extending from left one third of the transverse colon up to the mucocutaneous junction of the anal canal. So the mucocutaneous junction is marked by pectinate line. So up to the pectinate line, the alimentary tract develops from the hindgut. Also the mucous membrane, most of the mucous membrane of urinary bladder, urethra, then the parenchyma of prostate, parenchyma of bulbourethral gland, then the epithelial lining of the vagina. These are all endodermal in origin.